Hi, Michelle Glass here. Welcome back to our Chapter 10 lecture series. We're finally to the end of our discussion of how DNA molecules used to make proteins. We are ready for that second major reaction called translation. Remember, we can use our letter L to help us remember that translation is after transcription. Translation is the process where we take that mRNA sequence and we convert it into a protein. This is, of course, happening at the ribosome, which is our location for protein synthesis. This process of translation is going to incorporate or use all three types of RNAs: mRNA, tRNA, and rRNA. rRNA obviously composes the ribosome. This is happening at the ribosome, so that's how it's used. mRNA is bringing that gene sequence to the ribosome, so it's giving us the instructions. And then we've talked about tRNA as transferring the amino acids to the ribosome, so it's basically bringing our ingredients. All right, so here is our mRNA transcript that we made earlier in transcription, hence the name. And it's going to be the job of our tRNA to come in and read the mRNA transcript and bring the correct amino acid. And what we see is that the tRNA sort of like reads the mRNA in groups of three. And we call these groups of three bases a codon. So codons are groups of three bases that equal one amino acid, and our whole sequence is in these groups of three, and so we can call DNA molecule a triplet code. So it's describing basically how to break the code. Now, the tRNA molecule is fitting in based on the rules of complementary base pairing. So I sometimes like to talk about this molecule as having like hands and feet, and with its hands, it's holding up what we can call the anticodon, and with its feet, it's holding onto the amino acid. An anticodon is opposite to the codon, is complementary to the codon. So if our codon is AUG, then the anticodon would be UAC, and we just do that based on our rules of complementary base pairing. And that's essentially how the tRNA molecule knows the order of the amino acids to bring into place. Now, scientists have already figured this out, so we don't have to actually worry about those anticodons. Instead, we can use what's called a codon chart. And our codon chart here is already worked out, telling us based on the RNA, the mRNA codon, what amino acid the tRNA would bring into place. So you can see my first codon here is AUG. So using the chart, I can see that I'm bringing in the amino acid, which is abbreviated MET. Let's do our next one. Our next one is AAU, and so I am going to use my chart to see that our amino acid is ASN. So see if you can figure out the rest. Go ahead and pause your screen. Use the mRNA transcript I've given you. Use your codon chart. See if you can figure it out. So go ahead and pause. All right. Does yours look like mine? So I've up at the top separated my mRNA transcript into my codons. I've listed out the amino acids, and I've highlighted them in the chart. Okay. Basically then, if you have your chain of amino acids, you've got your protein. Remember, amino acids are linked together by special types of bonds called peptide bonds. So we can talk about this as a polypeptide, or we can talk about this as a protein. Now, the last thing to pay attention to here with the codon chart is you do have some codons that are described as stop codons. So these are going to be codons at the end of the gene sequence, signaling that translation is finished. And that's it.